Thank you. So it's great to know that my research accomplishments are only second to my Twitter accomplishments, which is definitely true. So thanks a lot for asking me to speak today and share sort of some updates from the lab. The majority of what I'm going to talk about today is unpublished, so it's really, work in progress, and so, I'd love to share this work with you. So what my lab has really been thinking about over the last few years is really this necrotic core. So we know that the necrotic core is sort of a feature of human plaques that we sort of consider to be the de facto most dangerous part of the atherosclerotic plaque, the one that sort of is prone to rupture and those kinds of things. But it's really actually a very complex process. And in fact, not a lot has been really elucidated in terms of the mechanisms that specifically contribute to the necrotic core. So when I first started my lab, this is something that I really sort of focused on and tried to hone in on some of these mechanisms. And cell death in the plaque is very, very important. It has a lot of actually beneficial roles in the atherosclerotic plaque. So during the initial phases of atherosclerosis, any cell that undergoes cell death, programmed apoptosis, will get engulfed quickly. And actually some of these things, most apoptotic cells in most tissues are very difficult to find because there's an immune cell right nearby that's there to clean it up and get rid of it. So in fact, when we're looking for apoptotic cells in any number of tissues, they can be actually hard to find, and that's a good thing. And apoptosis is necessary. It actually keeps the cellularity of the plaque down. Any macrophage or immune cell or vascular cell that has undergone stress, is cleared away, and this actually induces an anti-inflammatory response or a homeostatic response, repair response. And this is because the macrophages, the immune cells in the area, are primed and ready to engulf any of these dead cells. And they do so through a series of very conserved and highly specific receptors that recognize and clear these dead cells. What happens when this goes wrong? Well, there are two things. One is sometimes you can actually get an nephrocyte or a macrophage that is unable to engulf for a variety of reasons. They lose the ability to recognize these these dead dying cells. And then there's something that we've always termed secondary necrosis, and I'm going to get into this a little bit. And that was sort of always thought to be this sort of passive lysis of cells that just kind of, if they're not cleared away, they just kind of burst open and release all of their contents. And when a cell releases its contents, it's actually a very bad thing because all of those internal proteins, molecules, signals are all actually designed to be on the inside of a cell. And when the immune system sees those things at the outside of a cell, that triggers an inflammatory response. It signals there's a cell that has been damaged. So that's really where these two types of forms of cell death have sort of have diverged. We know that apoptosis involves caspases. It's programmed, it's controlled. We've all learned about this. Some of us in our advanced university classes. Some of us now in elementary school are learning about apoptosis. And like I said, that is absolutely required for tissue cell maintenance, any kind of injury, and requires the balance of an immune cell to clean up and to clear away the damage. Necrosis, on the other hand, releases these DAMPs, or these damage-associated molecular patterns. And those are the ones that I was just describing. They're really meant to be on the inside of a cell. They're not meant to be on the outside. And when they're found on the outside, they induce inflammation. And necrosis is always kind of thought of a passive process, like a cell is physically injured. You can imagine putting a cell or a tissue in a freezer and taking it out again. It might explode, or maybe treating acid or something on a physical trauma. But in fact, there's actually a cell pathway between the two of them, and that's termed necroptosis. And by design, the name is actually a combination of necrosis and apoptosis. It's programmed, but it's a necrotic type of cell death. It still releases all these damage-associated molecular patterns, so it is still very inflammatory, but it is very conserved, and it has a lot of molecular regulation along the way. So I'll walk you through the sort of the pathway specifically in this particular form of cell death that we became interested in. So a cell receives a signal from the outside in the form of usually something like a TNF-alpha, some sort of other kind of ligand on a death receptor. That'll initiate, often initially, a survival signal. The cell, an immune cell in particular, wants to stick around and fight that infection or fight that damage. But if for whatever reason those signals are impaired or the signal becomes overwhelmed, we know these prototypical caspases become activated and the cell undergoes apoptosis. But if happened to be that the caspases are inactive or other signals are there to sort of overwhelm the system, the cell can undergo a backup form of cell death, and that is necroptosis. This RIP kinase here that sits downstream of the TNF signal or the death receptor undergoes a series of molecular phosphorylation events and reciprocal interactions with other proteins, namely RIP3, MLKL. And this is all, again, tightly regulated. These are physical interactions between proteins. There are phosphorylation and dephosphorylation events, because as we might expect, you don't want a cell to undergo any kind of cell death, without a lot of control. But at the last step, this protein called MLKL comes in, and that actually inserts itself into the membrane. And instead of regulating an apoptotic form of cell death, this is where the membranes actually lyse, and all of the contents of that cell end up on the outside. And this is where the inflammation comes. So this is the type of cell death that is inflammatory. So then, knowing what we know about the way apoptosis may be interacting in the plaque and contributing to atherosclerotic plaque, we then wondered, does necroptosis actually contribute to atherosclerosis as well? And maybe that is why we see some necrotic core expansion in the atherosclerotic plaque. So this we did a couple of years ago, so I'm not going to go over this in detail, but what we found is that in human atherosclerotic plaques, either compared to normal arteries or in the stable versus unstable condition, we see these mediators, RIP3 and MLKL, on the gene expression level quite high. So they're higher than their healthy normal counterparts. So this, it might imply to us that this pathway is potentially favored in the advanced atherosclerotic plaques. I'm not going to go into the details, but we found actually that if you load up cells with inflammatory ligands that you find in the plaque, like oxidized or modified lipids, those cells can undergo necroptosis. So there are endogenous triggers of necroptosis in the plaque itself, and that they're regulated. This is in humans. And in mice, we can actually give a small molecule inhibitor of this process called NEC1 or necrostatin 1. And if we give it to mice that actually already have plaques, we can actually stop them from progressing. We can stop them from getting worse. And you can see here, this is all the necrotic debris that is found in a mouse atherosclerotic plaque. And you can tell just by looking at it that the cell kind of looks a lot different. There's not as much debris that there is when, in the mice that are not treated. So we can find then that maybe necroptosis is active certainly in mice. We can intervene, and it's sort of activated in humans. So it sort of implies that this might be a targetable, a druggable so-called pathway. We went upstream and we actually further tried to look to see whether or not RIP1, that first step of the signal for necroptosis, was was contributing to the atherosclerotic plaque. So we used siRNAs or ASOs, antisense oligonucleotides, to knock down RIP1 in mice. And once again, we can knock down the lesion. We can reduce the amount of lesion that these mice get quite significantly, actually. You can tell here, these are all these white chunky parts are all the lesions in a mouse aorta. And you could just tell, again, visually, that these are much reduced when you inhibit that first step of this signaling pathway. And there are fewer macrophages. The lesions are less inflamed. And if you actually look at one of the inflammatory mediators, IL-1 alpha, and IL-1 alpha is a really potent pro-inflammatory protein. And you look in the lesions of these mice, and there's much, much less IL-1 alpha in the lesions of the RIP1 inhibited mice. So really, no matter if we do it with a small molecule or if we do it with an antisense inhibitor, if we block this pathway, certainly at least in these mice, we're definitely reducing atherosclerosis. So this is kind of how we envision this part of the pathway. We think that initially, those inflammatory signals that are going on in the atherosclerotic plaque as it's growing are triggering RIP1 inflammation. That might be activating things like the NF-kappa B pathway, which is a really potent pro-inflammatory protein. The cell's surviving and promoting the inflammatory response, and then the atherosclerotic plaque grows. And what might then be happening is over time, there might be sort of an imbalance that's happening. So over time, we might actually be getting this, this secondary necroptotic activation that's happening, maybe in those advanced necrotic cores. So in both cases, we're able to intervene here, but I think, it might imply that the biology is slightly different, at early stages of atherosclerosis versus late. So, maybe in the future, this might be something that, as we're thinking about, therapies and what to target when, this might be, an example. But actually, MLKL, that last committed step, the step that actually ruptures the plaque and causes ruptures the cell and causes the inflammatory release. What about MLKL? It's sort of implied that all of this is happening, in the canonical, A equals B equals C. But in fact, we realized that, MLKL itself, we hadn't looked to see whether or not inhibiting the last step might actually be very efficient. And what we found actually when we initially looked in human plaques was that it's hard to tell, but these brown dots here signal the activation of MLKL. So these phosphorylated MLKL is that last committed step. And you can see that all of these activation events are really happening around the necrotic core, and this is in a human coronary artery. And when we quantify that actually across many arteries and many samples, we see that in advanced plaques, you see a lot of this phosphorylated activated MLKL, whereas in early plaques, you see almost none. And in fact, this is actually a very generous quantification. This is probably mostly just background staining from not very good antibodies. I don't actually think we can see much evidence of really, truly active MLKL in these early lesions. So again, it suggests that this protein might very much be something that we can target for that stabilization of the necrotic core. So then we think that if we knock down MLKL, we remove that stimulus for the lysis and rupture of cells, we can then, of course, reduce atherosclerosis and reduce necrotic core development. So this is Adil. While he's not drinking beer, he's spending a lot of time injecting mice and looking at atherosclerotic lesions. So once again, we use these antisense oligonucleotides. These are great, actually. They're from a pharmaceutical company called Ionis. And Ionis has a lot of clinically approved antisense oligonucleotides, mostly targeting the liver, so nothing really in the plaque yet. But they get to the plaque, they're very safe, and actually it means that if we can find something like this using one of these antisense, we might be able to actually move it into the clinic actually quite rapidly. So we just did a typical thing we do in the lab all the time. We take these mice that get atherosclerotic plaques, and we gave them this antisense oligonucleotide or a control that doesn't target anything, so just to kind of give it as somewhat of a placebo. And like we suspected, when we inhibit MLKL, we actually do see reductions in the necrotic core. So similar to what we saw with that necrostatin molecule, we're getting what we expect. But unlike what we expected, surprise, the plaque area actually didn't decrease. So this inflammatory cell death that sort of we think is kind of perpetuating the inflammation and contributing to the plaque growth didn't seem to be stopping the plaque from actually getting any bigger. And it certainly wasn't any smaller. It actually trended to getting a tiny bit bigger. So we looked a little more carefully. Maybe MLKL, maybe our antisense wasn't doing what it was supposed to do. Maybe it didn't inhibit the necrotic death like it was supposed to do, but it did. So these little green dots are all these apoptotic cell death, cells undergoing cell death. We don't know what kind of cell death they're undergoing, but they're green dots implying dead cells. And you really don't see, you see very few when you inhibit MLKL. So you can also tell you see very few when you don't inhibit MLKL as well. So it goes to show you in my initial, when I first was explaining about the phagocytosis, that's a really important part of the atherosclerotic plaque is the phagocytosis of this. But anyways, we don't see any MLKL, we don't see any cell death, very little cell death when you block MLKL. So it seems that MLKL inhibition is doing what we thought it should. Well, then why aren't the lesions any better? Why aren't they less inflamed? And what else might be going on? Turns out the lipids in the plaque, so the red stain here is oil red O, and it just marks a cholesterol or a neutral lipid in the plaque. There's a lot more of it. So we were very surprised by this. In fact, we tend to do oil red O as kind of a one-off to make sure that the lesions are what we think they are. We don't, often usually they correlate with lesion size kind of very well. And sure enough, the oil red O area in these mice treated with the antisense to knock down MLKL was much bigger. When we looked in vitro, we saw the same thing. So you take macrophages out of an MLKL-treated mouse, an ASO-treated mouse, and you can see here by various stains for lipid. The macrophages here, outlined here, contain more lipid. They actually have greater number of lipid droplets, and the lipid droplets actually contain more lipid. So it's something that's going on in the plaque that we don't fully understand, but there's something going on with the way that MLKL controls lipids. So we started to investigate this a little bit further. And this is the kind of thing where you encounter unexpected results in the lab, and you can either stop and not think about it, which is what I do a lot, or if you're Adil or other great people in my lab, you kind of go, no, let's try this. Let's try, let's see what's going on. So this is what he did. So we tried to look, how does MLKL then influence a macrophage? So here's a cell death protein that's supposed to lyse the cells, and now we're finding that when you don't have that protein, the cells are actually, all their lipids are backing up. So maybe they can't efflux their cholesterol. Maybe they can't get rid of their cholesterol. Well, they can. They can efflux, no problem. Well, maybe they're taking up more cholesterol, at the beginning. So maybe when you feed a cell with, the absence of MLKL, it's actually, engulfing more, and that's actually why. Well, that's not true either, actually. We found that there's no difference in the amount of cholesterol uptake or the amount it gets rid of. And we found that it doesn't interact with any of the proteins that normally regulate cholesterol uptake, such as CD36 or ABCA1. So there's a lot of things that MLKL does not do. And that only ever boils down to sort of one slide or half a figure in supplemental. But that you spend a lot of time finding out what something doesn't do. And this is one of the examples. Well, it turns out when a cholesterol molecule is taken up, it needs to be trafficked through a number of vesicles throughout the cell in order to actually be metabolized. So the cell, can store lipid, but then it can actually mobilize it through various sources. And what we found is if you look at MLKL expression, and that's just in green, and the various markers of these vesicles, RAB7 being the late endosome and CHIMP4B is a marker of something called the multivesicular body. We see that MLKL is found in these vesicles. So it's not at the cell membrane lysing the cell all the time. It's doing something else. It's actually trafficking vesicles to where they're supposed to go. And when we knock out MLKL, basically all of this gets messed up. The cell cannot process these vesicles like it normally can. So MLKL is then needed and required to move the amount of multivesicular bodies or these other markers of other vesicles through the cell efficiently. And when you don't have MLKL, all of that gets stuck. And then the lipids get stuck. So when you get lipids accumulating, they get stuck in these vesicles and they can't get out. So essentially what we're thinking is that MLKL is really allowing the cell to process the amount of cholesterol that it takes up, and that in the absence of MLKL, the lipids get stuck and the plaque then potentially grows. So what does this mean? This means that the cell is ultimately, in the body, first of all, MLKL is doing far more than we thought. It's more complicated. It's not simply a necroptotic, protein that's lysing cells. It tells us also that, my certainly intention to sort of jump into these, we can therapeutically target this, that'd be so cool, imagine. Well, it really needs the biology behind it, because until we understand what these proteins and these molecules are actually doing in the cell, we can't intervene and just assume that we can inhibit it and only one downstream pathway is going to be affected. And it also implies that in this case, the cell is really under constant pressure to survive or to die. And it means that it needs to react to its environment, in this case in the atherosclerotic plaque, it's in the phase of excess lipids, but it also needs to understand that at times the excess lipids are too much, the cell membranes are damaged, the proteins are damaged, DNA might be damaged, and the cell needs to die. And we think MLKL is kind of sitting at this intersection. It is sitting, understanding what is coming in from the outside with respect to cholesterol and how then it can mediate cell death. And this is, and we're still learning a lot about this process, and by no means, is this solved. But looking a little bit deeper, we noticed two other very interesting things when we inhibited MLKL. One, so as I mentioned, the necrotic core is decreased as we expected, the lesions are smaller. The other thing that was much decreased was plasma cholesterol. So what is a protein that's mediating cell death in the plaque doing regulating circulating plasma lipids in the animal? And skip to the end, we have no idea. But it's happening, and it's very complex. The other thing that we noticed, and this is actually something, again, where one has to have their eyes wide open when they're doing these studies, is the spleens of these animals were massive. So this is, for those of you not used to looking at a mouse spleen, this is what a mouse spleen looks like. This is what a very large mouse spleen looks like. They're very, very, very expanded. So there you can see here, they're almost double in size. So what is happening in the spleen? Well, when we look a little bit closer, the spleen is an active area of hematopoiesis. It's where the immune cells can mature. It's actually an immune organ. And we actually look at these regions of immune cell development in the spleen. In the absence of MLKL, you can see all these blue dots are immune cells. So there's a sort of an expansion of immune cells in the spleens of these mice, and you can see that here quantified. And a few years ago, a number of groups, including Matthias Nerendorf and Phil Swirsky in Boston, discovered that the spleen actually holds a reservoir of monocytes that become macrophages that contribute to atherosclerosis. So the spleen is not sort of a passive organ. It's not just a backup immune space. It's actually constantly providing a source of immune cells that specifically go to the atherosclerotic plaque and expand it. So maybe this is why we're seeing, the expansion of the atherosclerotic plaque, or the lack of expansion, or the lack of protection of the atherosclerotic plaque when we have MLKL, in the absence of MLKL. So hematopoiesis goes like this. The bone marrow is generally speaking the main source of hematopoiesis in the body. So there's a vascular niche, there's a whole bunch of progenitors in there, and when needed, those progenitors can mobilize and become the various types of immune cells. There's the common lymphoid progenitor, which becomes the T cells, the common myeloid progenitor, which becomes the megakaryocytes, platelets, macrophages, monocytes, and neutrophils. So when under certain forms of stress or under various conditions, the spleen can essentially be a source of all of that. So we wondered then if maybe MLKL might be playing a role in this process of extramedullary hematopoiesis, so basically outside of the bone, hematopoiesis that occurs outside of the bone. And we find that if you actually look at endogenous MLKL expression, this is actually in humans, this is basically just a measure of the RNA, the levels of number of copies of MLKL RNA in these various cell types. And you can see that these hematopoietic progenitor cells, so HSCs, the red pulp, monocytes, NK cells, there's all of these immune cells that express a huge amount of MLKL compared to other forms, compared to T cells, compared to B cells, that kind of thing. So it doesn't tell us anything. Of course, association doesn't mean causation, but certainly the levels of this protein are very, very high in these various immune progenitors. Maybe that's something that hints to us what's going on. When we actually do a stain and we look at where MLKL is actually expressed in the spleen, it marks this red pulp quite well. So the red pulp is an area where we see the monocytes develop, and the white pulp is where other immune cells develop. But really, the red pulp is expressing high levels of MLKL. But when you put mice on a Western diet, so you raise their plasma cholesterol levels, and this is meant, of course, to mimic the atherosclerotic environment in humans, we lose MLKL expression. We don't know why, we don't know how, but we lose it. And that may correlate to why we see an expansion of these monocytes that contribute to atherosclerosis under the same condition. So perhaps MLKL is acting as some kind of brake on the hematopoietic expansion and the immune cell mobilization out of the spleen. And when you don't have MLKL, we see an expansion of those immune cells. So we asked whether or not MLKL influences extramedullary hematopoiesis. So for this, Adil took mice where MLKL is once again inhibited using the same antisense approach. And essentially what we do is we can ask whether or not the progenitor cells from the bone marrow or the spleen form the colonies that they're meant to form. So from a progenitor, can it become a myeloid cell? Can it become a macrophage like it's supposed to? We can look at this by flow cytometry, and we can look at also in the peripheral blood to see if this is affecting the peripheral blood. And I won't go over the millions of dot plots that are involved in looking at the flow cytometry because, frankly, I don't really understand it. But I will say that the conclusion is we see a significant expansion of every form of the myeloid progenitor population in the absence of MLKL. So indeed, in those spleens that are massive, there are more of these progenitor cells, and of all stages of the progenitor. But interestingly, when we did the same experiment, we looked in the peripheral blood and the bone, and we did not see the same thing. So it is not that MLKL is just globally regulating all immune cells. When you don't have MLKL, you don't have like a leukemia, for example. But you do have a very specific expansion of these macrophages that come from the spleen. And we know that those are particularly pro-atherogenic. And when we look at this in culture, we see something kind of similar. You can see that these are our splenocytes that we've put in culture and looked to see if they form colonies. And you can see in the absence of MLKL, the only difference between these two wells is really the absence of MLKL. And you can see, quite an expansion of the number of cells. So then what we think is happening is that MLKL is regulating when MLKL is absent. There's an expansion of these progenitor cells in the spleen. We still don't understand why. And it turns out nobody really knows what the niche is in the spleen that might be affecting the way progenitors develop. We know a lot about the niche in the bone. We know a lot about the way that the hematopoietic progenitor cells interact with, say, the endothelial cells or the stromal cells, the fibroblasts in the area. We don't know a lot about how this is happening in the spleen. Some think it's the same. It's just the same niche in the spleen as it is in the bone marrow, but we know it's not. We know the spleen is not identical. It cannot replace your bone marrow. And then once MLKL is gone, whether it be in our model where we've knocked it out, or when you maybe have a high-fat diet, high cholesterol diet, and you see reductions, these monocytes potentially enter the circulation and contribute to the atherosclerotic plaque. The plasma cholesterol part of it, we don't know. And in fact, there are models where if you see expansion of immune cells, so in various leukemias, for example, you actually do see a reduction in plasma cholesterol because the immune cells are actually eating up all the cholesterol in the circulation. In fact, when you think about it, cholesterol is an energy source. And so when a particular tissue needs energy, it's going to use it. So it's possible that the immune cells that are expanding in the spleen are just kind of sucking up all that cholesterol. And actually, as soon as 20 minutes ago, we were taking out these spleens with the help of Rick and seeing whether or not that actually normalizes the plasma cholesterol in these mice to see whether or not that's the case. In the circulating immune factors, we're also finding a bunch of different proteins that are different when MLKL is gone. So, in addition to how we are seeing it in the regulation of plasma cholesterol, in the regulation of the hematopoietic cells and the plasma cholesterol, it's also regulating immune factors. So we really think again that MLKL is really sitting at the intersection between the metabolism and the immunity and the inflammation. So thinking then on the other side of the coin, and that's the efferocytosis of dead cells. So that clearance that is absolutely necessary. We turn over something like 100 million cells a day in our body. And as I said, it's difficult to find them. If you look in certain tissues, you don't see any. It doesn't mean there's not apoptosis, but it just means they're being cleared. And as I mentioned, this involves a sort of tolerance, an anti-inflammatory, pro-reparative, an immune silencing essentially. It involves TGF-beta, synthesis of collagen, really anti-inflammatory cytokines. But if you don't have efferocytosis, so you do not have the clearance of these dead cells, there are a number of questions about what happens. So as I mentioned, some of this might be passive necrosis. So does the cell just kind of start to dissolve? What happens to those dead cells if you don't clear them away? And is that actually a bad thing? Are we actually contributing to the atherosclerotic plaque in that way? So we set up an experiment and we had a hypothesis, and this is Claire in the lab. I always like to put pictures of my trainees having beer because it makes us all seem, fun and crazy when we're not in the lab. And Claire revealed to me that this is actually pineapple juice. It's not beer, but that's okay. It goes with the narrative. So what we can do, we can actually, in culture, we can block efferocytosis. So we can take all of the receptors that the cells have for efferocytosis, and they have a lot. They've got a lot of redundancy. The immune system does not want to leave a dead cell behind. So it will have a lot of various types of ways that it tries to recognize a dead cell. But we can block that. We can take a macrophage, a normal healthy macrophage, block efferocytosis. And these are green cells, and I'll show you that in a second. And we can feed it dead red cells. We can actually just add the dead cells that are labeled red to the macrophages and then count how many cells are engulfed by the macrophages and how many are not, and then what happens next. And you can see here in a normal condition, the green macrophage has eaten up a red macrophage, a dead cell. There's a little one here on the outside, but there's sort of two potentially on the inside. When we block efferocytosis using these various receptor blockers, these dead cells just kind of stay on the outside of the macrophage. They cannot be engulfed by the macrophage. And we see that here. So it's a reduction of usually about 50% reduction in efferocytosis or the phagocytosis. Note that we don't ever really get 100% reduction. So it's really hard because of the redundancy. So we're constantly having to block using these multiple receptors because the cell has really evolved. We've really evolved to be able to engulf macrophages really, really easily. And what happens to those cells? So if we take those cells that are not engulfed and we actually assess them as dead cells, what do they look like? Do they stay dead? Do they passively acquire this secondary necrosis? Are they just bursting, just sort of, as we thought they might? And what we find is actually that, so this is a measure of necrosis here on the y-axis, and we see that over, with these different blockers, you see a lot more cells, adopting this necrotic phenotype. We can quantify that here. But actually what's very interesting is that those necrotic cells actually have that phosphorylated MLKL, that marker of necroptosis on their cell surface. So it's not just passive necrosis. It's not just the cells, dissolving their membranes passively. It's actually active. And you can see that here by Western blot as well. When we block efferocytosis, we can see here that we see a marker of that phosphorylated MLKL. So what does that mean? Why does it matter? Why does it matter whether or not we can block, we can enhance efferocytosis or we can block necrosis? Because it means it's active, and it's active in a way that we can control. So now, I've just told you that MLKL is too confusing to be able to target it, therapeutically, but at the very least, this tells us that both at the upstream level, as the cell is undergoing death, we can really block it at multiple time points. And even in an advanced plaque where efferocytosis is maybe impaired because of various reasons, and we know a lot about the way that efferocytosis is impaired, it might be that we can rescue some of this by actually blocking MLKL or blocking one of the other ways that the cell recognizes these cells. And we can show that actually, this was kind of hard to put together, but these little dead cells here are in green, and what you actually see is that when you don't have MLKL, macrophages are able to phagocytose all those dead cells a lot better. So we can show in the plaque itself that the phagocytosis of those dead cells that are not undergoing necroptosis is much better. So it's another reason potentially why this pathway is really important. So what's next? We've been thinking about the necrotic cell death quite a bit. One of the pathways that I'm not going to tell you about today, but we've actually been able to find ways to interfere with this. So there are anti-inflammatory lipid mediators that are produced during the resolution of inflammation. And it turns out one of the pathways that they hit is this RIP1, RIP3 necroptosis pathway. So it means that during the resolution of inflammation, it's possible that the immune cells are actually disarming this ability of the cell to undergo inflammatory cell death, which of course means, once again, it's potentially druggable. I'm not telling you about this today, but the sort of other half of my lab works on microRNAs. Unsurprisingly, a cell that is constantly undergoing a decision whether or not to undergo cell death or survive, it's using these microRNAs to decide. So we think that under certain stimuli, microRNAs are sitting there and inhibiting particular transcripts like MLKL, like RIP3, so that the cell cannot undergo death. And then when the time comes, that's released, that brake is released, and the cell undergoes cell death. And of course, a biomarker. Can this be used as a biomarker? So we've been thinking about this for a while, and we've actually able to detect some of these pathways, some of these proteins in the plasma of that in higher patients with CAD. Others have gone on to show the same thing. I'm not showing the data here, but we did actually work with Terry Ruddy and the imaging group here to develop an imaging molecule. It was a SPECT molecule, but it could also be a PET molecule to be able to detect this pathway in atherosclerotic plaques, and we did that in mice. So it certainly means that I think this pathway, the more we learn about it, the more steps that we fill in between, we learn about the way that this process actually happens and how we can intervene. So I just want to thank the members of my lab who did this work. I already mentioned Adele and Anne-Claire. Anne-Claire has gone back to France. Where is Anne-Claire in this picture here? There she is. She's already gone back to France, but actually she's coming back to do some more work, and so we're excited to have her. Michelle, who does basically everything in the lab and takes care of all of us, and when she went away to have this very small person, we nearly fell apart, but we managed when she got back. Mary Lynn, who works on the microRNA side of things. I didn't talk about the project that Leah's working on, but she is similarly working on how mitochondria are at the center of all of it. So mitochondria are very much the energy sensors and in a lot of ways, the immune sensors of the cell. And so how do the mitochondria actually react to their environment? As well as Mayanne just recently left my lab, and now she's with Aaron Mulvihill. But Mayanne has worked on many of the microRNA pathways as well that I didn't talk about today. And we're now sort of working even more, with others here at the Heart Institute and elsewhere to figure out, other regulatory pathways. Mireille Ouimet and I are submitting actually a grant in 48 hours about how these pathways are impacted by aging and how, the aged environment, actually a lot of these sensing pathways and a lot of the detection and clearance pathways are impaired. So that's where we're going now. So thank you very much to everybody, and I'm happy to take questions.